I'm really pleased in both cases to make an introduction here. And those of you who don't know, we have a rich history, actually, pretty rich history, not monetarily rich, but rich with traditions here at the Marine Sciences Research Center originally, and now SoMAS, School of Marine Atmospheric and Sustainability. So one of our key professors here in the initial period of the Marine Sciences Research Center was Okubo, and he was an esteemed professor of first chemical oceanography, then physical oceanography, and then subsequently mathematical ecology, which he's extremely well known for. And so, in fact, there's a dedicated volume of the Oceanography magazine of the Oceanography Society from 1999, celebrating his contributions to education and research over the years. And so now, of course, I can't. Okay, so you can read something about his bio here, but one of the things that you can get from this list is that Jerry Schubel, who was really our first dean and director, Akira was his first appointment to the faculty as in his role as such. And Akira came in 1975, and you can see he was in those days he was actually extremely productive. Nowadays, people publish a lot more in group publications, so maybe they have hundreds of publications, but his publications were very impactful. And in fact, in the physical oceanographic area, when I was actually an undergraduate student, I read this paper by Okubo on oceanic diffusion diagrams. And one of the things that he often did was relate scale, spatial scales or temporal scales in the environment to some factor, in this case, of dye dispersion in the ocean, measured as eddy diffusion coefficients. So eddy diffusion coefficients increase here, scale of the features that you're examining increase here, and you can see that the bigger the scale you observe this process, the bigger the process seems to be. So he made many contributions like that, and one of his contributions here was to our graduate education program. And every Friday afternoon, he used to participate and lead a seminar that was a wide-ranging seminar that graduate students and professors participated in, and then afterwards we had beer. And one of the people that he mentored and participated in the seminar was Greg Marshall. Some of you may know Greg Marshall. He invented the critter cam, which you see on all sorts of programs now and used in ecology. And so he was a very important person in that regard. And he published a highly influential book called Diffusion and Ecological Problems, Mathematical Models, and was well known for that contribution. And he also, one of the characteristics of Akira was that he loved to derive simple analytical models to describe physical processes. And he did this often in just absolutely beautiful script, mathematical script form, which has kind of gone by the by as numerical models have taken the primary position. So he followed up with the second edition, written together with Simon Levine, an extremely well-known ecologist from Princeton. And this was from Malcolm Bowman. I put it in this slide to show, again, another one of his scaling diagrams, the scale of observation here, or the scale of the thing that you're observing, whether it's plankton or a dragonfly larva or whales versus the swimming Reynolds number. So he made a huge number of these contributions. He was very influential, and we had a symposium for him in 1995, shortly before his death in 1996. And the speakers at that symposium were like top people everywhere in the country. And that shows you the esteem that Akira was held. I mean, he was a foremost individual in this area. We had Simon Levine, that I just mentioned, people like John Steele, Edie Cole from Berkeley, came, Trevor Platt. These are names that are huge figures in the field of mathematical ecology and oceanography. Kira also was he was an interesting, funny guy too. He had a real sense of humor. Here's one of his favorite restaurant in Port Jefferson was Hana, a Japanese restaurant, as you might guess. And here's a map, directions to Hana. You can see here things like to Asia here, to Africa, to Europe, and so forth. And then Lock Island Sound. I don't know what the heck he was thinking, but he had a real sense of humor. And so I'll just end by saying thank you, Kira, and one of our figures here that helped form our school. So today we're going to have in his honor, like those other famous people I just showed you, we have in his honor brought other new famous people, younger famous people, present famous people, and David Fike is one of those. David is another esteemed and influential person, and I'm really delighted to introduce David Fike, who's another, as I said, famous person in a limitless line of famous people I just mentioned. He's presently the chair of Earth, Environmental, and Planetary Sciences at Washington University, St. Louis. So a very fine university. He's a distinguished university professor there, a named distinguished university professor. Now, and he did his PhD work, by the way, and many of you heard yesterday because he gave his career background yesterday. He did his PhD at MIT, and he has following on a line of intellectual development there and extending it. His expertise is in the biogeochemistry of particularly the lighter elements using isotopic patterns, as you're going to hear more about today. Now, I met David many years ago at the University of Chicago, not when I was still there on faculty, but we were both happened to be there to give seminars. So he was giving his seminar, and then which I heard, and I thought, that's fantastic stuff. I gotta meet this guy, and it's really been great over the years to have built on that relationship. So I'm really pleased, David, that you were able to come and participate in this context of the Okubo Scholar, and please lead the way.
Thank you. Thank you, Bob, for that lovely introduction. It's a real pleasure to be here as the Okubo Scholar. Big shoes to fill, and I will do my best. I am going to talk today about integrating bulk scale geochemical measurements and micron scale measurements, and how we can gain understanding into process and mechanism by integrating those things. I'm going to talk specifically about pyrite today and the sulfur isotopic composition of pyrite. But I will try to make the case that these principles are pretty broadly generalizable to a range of elemental and isotopic approaches to understanding modern and ancient systems. I want to start with some acknowledgements of collaborators, including Bob, who has definitely shaped my thinking on this since our fateful meeting in Chicago many moons ago. And then particularly collaborators at the Weizmann Institute in Israel. Itai Halevi is really a leader in biogeochemical modeling. And then Roger Bryant, who was my PhD student, and he is now on faculty at Purdue, and funding sources that make all of this stuff possible. There's Roger smiling here. Sort of our little lab group. And again, these are the people that make the magic happen. I'm a geobiologist. For those of you that were here yesterday, I sort of went into my background in a lot of detail. This is the phase space that I like to think in. I spent my PhD time really looking at the global scale and my postdoc time looking at the small scale. And at WashU, my research really trying to link these things together. And we do that by looking at metabolism environment interactions. We do that by looking at how depositional conditions influence how those geochemical signals get preserved in the rock record. And then we do that by trying to clarify and understand how post-depositional processes can alter those geochemical signals, how to identify that alteration and sort of subtract that from what we're trying to see. And I am a stable isotope person. That is the tool that I use to try to extract this information. And the reason for that is that those biological and abiotic reactions often show a discrimination or a preference for one isotope versus another. These preferences can be really quite large relative to our analytical precision. And so they're really strong levers with which to investigate these kinds of processes today. And then because these isotopic compositions can get preserved in organic matter and in sedimentary minerals, they provide an archive over Earth history that we can use to reconstruct this long-term evolution. We're going to geek out on sulfur isotopes today. Sulfur's got 4 isotopes that are stable. We're going to just talk about the 2 major isotopes, 34, 32, and this ratio here, delta S34, that looks at that ratio relative to a standard. The canonical way that we think about the sulfur cycle is sort of inherited from Bob Berner at Yale. The ocean is full of sulfate. We know the isotopic composition of material that enters that ocean, and there are 2 pathways predominantly that sulfur leaves the ocean as sulfate minerals, gypsum or anhydrite, as well as barite, or sulfate that gets incorporated into carbonates, so carbonate associated sulfate. That's over here, and this has the same isotopic composition as seawater or something very similar to it. The other pathway is following the microbial reduction of sulfate to hydrogen sulfide, which then can interact with iron to form pyrite. Can also interact with organic matter to form organic sulfides. And because of this microbial sulfate reduction, there's a large isotopic difference between these 2 sinks. And so the prevailing framework is then that depending on which of these sinks dominates, that controls the isotopic composition of the phases that leave the ocean. So for a constant offset between these phases, the isotopic composition of sulfate or of pyrite both linearly increase with the fraction of sulfur that leaves the ocean as pyrite. So this would be all the material leaving as sulfate or all the material leaving as pyrite. And then what people do is they make measurements of sulfate or pyrite, and then they infer this pyrite burial flux, or the ratio of sulfur leaving as pyrite relative to the total flux leaving the ocean. Because these are strongly different in redox, this is one of the dominant controls on Earth's surface redox. And this kind of framework, I mentioned, was sort of pioneered by Bob Berner, but it is used by a whole slew of people to try to reconstruct how Earth's redox system has changed. This is true for the Cambrian, this is true for ocean anoxic events, and also trying to reconstruct things in the Ediacaran associated with the rise of animals. I'm going to try to make the case that all of these people, especially these guys, were wrong, that they had no idea what they were talking about. And so that this is the framework that people have. You get stratigraphic records of, in this case, carbon isotopes and sulfur isotopes. You get really excited. You take that to your collaborators and you tell a story about temporal variations in ocean chemistry. Right, so what we do is we take a stratigraphic record of isotopes and we assume that this reflects a temporal change in ocean chemistry. And that's very appealing, that's very sexy, that's how you get a lot of science and nature papers. Right, and so in this context, in that Burner-esque model, this is a pulse of organic carbon burial, this is a pulse of pyrite burial. This has profound changes in oxygen concentrations in the atmosphere. Or, and I hope I can convince you, this kind of pattern is caused by something else. Specifically by changes in local sediment deposition conditions. And just as an example, in this case here, this is the SPICE event, the Cambrian. This is associated with a massive regression, transgression cycle. So major changes in deposition that tends to get left out of some of the geochemical interpretations. Right. And one of the frameworks for thinking about this comes from a study that Bob did 2 years ago. And it was looking at fairly shallow cores in the Gulf of Papua here. And we went and we looked at the pyrite isotopic composition preserved in these cores, going from about 8 meters of water depth to 50 meters of water depth. The very depleted values, the deeper cores tended to have very depleted values, not very variable. The shallower cores all had heavier values that were a lot more variable. And so this was like the key piece of evidence that said deposition really matters. That the environment where your sediments are forming matters a whole lot. That was probably obvious to people like Bob, but for me, it was a real revelation. And the reason why that matters is because these pyrites are forming in the sediment. They're not forming in a well-mixed water column, they're forming in the sediments associated with ongoing microbial processes that draw down seawater sulfate into the pore waters, producing hydrogen sulfide. Because of that large isotopic fractionation associated with sulfate reduction, this results in a distillation of isotopes for the residual sulfate and a corresponding enrichment in the hydrogen sulfide that gets produced. This is a ubiquitous sort of phenomenon. This is a compilation of data from Claypool looking at sort of sedimentation rate here and the sulfate reduction rate, which is associated with increased values here. You see that there's this really strong correlation towards heavier values, faster rates of sulfate reduction associated with enhanced sedimentation rates. And so this is that's not what that's supposed to look like at all. Interesting. All right, we're going to go with it. Imagine that this is a sequence stratigraphic context of sort of a high stand, low stand, high stand deposit. Here I've color coded those with the isotopic composition of pyrite that we could predict just based on things like sedimentation rate. And what you can see is you get very different values. If you were to take a geological section or a stratigraphic section and then try to analyze those, the values that you would get at different places would look wildly different in terms of the stratigraphic expression of those isotopes. So this is a sort of a conceptual schematic. We then went and tested this looking at a core just in the Gulf of Lion off the south of France. And this is Virgil Pasquier when he was a PhD student. And this is looking through recent glacial interglacial cycles. So the last 500,000 years. In orange here is relative sea level change. So you're going through glacial to interglacial cycles. And the data in black are pyrite on the axis on the right. And the pyrite axis is flipped. So negative values are at the top and more positive values are at the bottom. You can see that there's a really strong correlation between fairly negative and at variant values during interglacial periods, and then more enriched values during the glacial. This can be seen in this histogram very nicely. These are the interglacial values. Pyrites are very depleted, not too variable, and then the glacial settings where they are more enriched and a lot more variable. Here, these are also color coded by reconstructed sea surface temperatures based on organic biomarker proxies. So warmer values tend to be here on the left, cooler values there, right. And so there's a strong difference between glacial and interglacial values for pyrite isotopes. Right. Well, this is what we see: high values for pyrite isotopes during glacial times, where we're shallower water, more close to shore, more detrital input. Lower values, less variability when they're deposited in interglacial times, farther away from the source of detrital input. There are a variety of different mechanisms that could explain this. One is biological. One is there's a change in the fractionation that microbes do during sulfate reduction. Specifically, faster rates of sulfate reduction during the glacial times and slower rates during interglacial times. And the data to support this is laboratory experiments here, where the x-axis here on a log plot is cell-specific sulfate reduction rate. So how fast those microbes are metabolizing. And the vertical axis here is the magnitude of isotopic fractionation, the separation between sulfate and sulfide during that activity. So if they metabolize very slowly, there's a large fractionation. If they metabolize very quickly, there's a small fractionation. So the way I think about that is I love M&Ms. I love red M&Ms particularly. If I get a bag of M&Ms and I eat them very slowly, I will take all the red M&Ms and leave all the other ones behind. The way I usually eat M&Ms is I just shove them in by a fistful. And when that happens, there's no separation in M&M colors between David and Ben. So this is how I normally eat them, very fast, no fractionation, versus my ideal way. There's another possible explanation. And that has to do with the sedimentary environment, that we are switching not in microbial processes, but in sedimentary processes, going from a more open system to a closed system. And we see evidence for this as well. And again, this is the framework here, where in interglacial times, we have pyrite that's forming here very early, near connection to the overlying water column. During the glacial times, we would have pyrite forming deeper down, where it's more isolated from the overlying water column. What evidence do we have to support that? This is a plot of sedimentation rate on the x-axis versus the isotope composition here. Again, sort of color-coded by glacial, interglacial stages. And you can see that there's a general trend towards heavier values associated with elevated sedimentation rates. And that makes sense. If you have higher sedimentation rates, you're more efficient at isolating your pore waters from the overlying water column. So we have two possible mechanisms to explain this. There's a third mechanism I didn't talk about. That is, there could be a change in the parent composition of the sulfate, but the sulfate reservoir is sufficiently large, and glacial interglacial changes happen so quickly that there's no change in this pool. This is one of the reasons we looked at the Pleistocene. We could rule that out. And then we're left with these two mechanisms: a change in biology or change in sedimentation. How do we distinguish these? So I'm going to make the case that we can distinguish these by looking at the suite of pyrites that form. Rather than looking at that single bulk value, look at the value they all form. And the reason that we can do that is we would expect very different histograms of the isotopic composition of those pyrites in different cases. This is kind of a busy diagram. In the interglacial case, this is just looking at the drawdown of seawater sulfate. Pyrites are forming early on in a fairly open system, and this results in a very narrow distribution, the isotopic composition of the pyrites that form. An alternative, and that's really the only way to get that invariant interglacial value. In the glacial settings, we have these two contrasting methods. One is we've just changed the biological fractionation here. In other words, as sulfate is being drawn down, there's just a change in biological fractionation. Nothing else is really happening. Isotopically, that just means there's much less enrichment in sulfate and a smaller offset between sulfate and sulfide. Otherwise, a pretty open system. That would produce a histogram of pyrites that look very similar to the interglacial ones. They're just shifted to higher values because the fractionation, the offset between sulfate and sulfide is different. Alternatively, you're drawing down your sulfate pool and you're forming pyrite deeper down where it's got more distillation happening. That's shown in these dark blue arrows here. And in that case, you're starting out with very depleted values and then getting a tail to very enriched values. So your histogram would look something like that. Huge variability in the values that we get for pyrites. Right. So how do we distinguish these? Again, we go on with the SIMS and we make individual measurements across all of these different data points. This is the schematic for how SIMS, secondary ion mass spectrometry, works. You load your sample into a high vacuum chamber and bombard it with primary ion beam that's focused. Here we use cesium. We can focus it down to about 0.5 micron in size. We raster that over the sample area, projecting secondary ions, which we then focus onto a series of collectors. People have been using SIMS to make measurements for of pyrite for a number of years. This is the instrument that we use in my lab. There are a couple of different ways that one could use these instruments. This is some work that I did when I was a postdoc. This is looking at Archean pyrites, and here we're analyzing things in spot mode, where you basically zap individual spots, and then you try to infer spatial patterns based on those. And that's one way to do it, but it's pretty coarse. These are 25 micron spots, so you're really not able to see details. The way that we do this now is using ion imaging. So instead of a single spot, we raster our beam over the target of interest, and that records the spatial information. So this is a framboid, a lovely little pyrite framboid. And this is the isotopic values that we can get for those individual framboid microcrystals. So we're able to have a much more refined sense of inter and intra-grain variability. And this is the approach that we use to move this thing forward. This is a plot. This dark line with the X's is that glacial interglacial bulk data that Virgil produced as part of his PhD. These box and whisker plots are the SIMS data for those bulk samples. So each little dot here is an individual pyrite that we've analyzed. And in general, what you see is that the SIMS average values agree or are offset slightly from that bulk value. The minimum value for all of these is about the same. It's about -50 per mil. In the interglacial values with low bulk values, there tends to be less variability. In the case of the glacial samples, we have a lot more variability with some values getting up to +100 per mil. So this is consistent with the idea that we have closed system distillation driving these changes in bulk values. Everything starts out the same, and your bulk value is controlled by the degree to which you have closed system distillation. That's exactly what I said there. We want to see sort of how widespread are these patterns. So instead of the last 5 glacial interglacial episodes, we moved a little bit further back to the mid-Pleistocene and went to the Crotona Basin in southern Italy, looking at the mid-Pleistocene transition 800,000 years ago. This is what the bulk pyrite record looks like through this interval. It is generally depleted in interglacial times, gets enriched in glacial, depleted interglacial, enriched glacial times. And again, we can constrain what we think are the relevant parameters for these kinds of changes, things like relative sea level, TOC, and sedimentation rate. Using just those values, we can predict what pyrite isotopes should be. And that's what this line here is. It's the prediction value here in black overlaying on that bulk value. So we can do a pretty good job of using generic sort of sedimentary parameters to reconstruct what that bulk value is. And that's just assuming we're going from open versus closed system. There are some real outliers, though, that can't be explained by this. So there are secondary processes that have to happen. And those secondary processes we can gain insight into by looking at the histograms of these individual pyrites. So these are just 5 of those samples. And these are the histograms of those individual pyrites. And I'll talk through those in more detail in a second. One of the really nice things about using this kind of approach is we get petrographic information about all of these pyrites. So we can see if we're dealing with nice rhamboids or aggregates of microcrystals. We can see if we are dealing with some euhedral pyrites or things that appear to have undergone some recrystallization or overgrowth textures. And we can then sort of color code these when we look at what happens isotopically. Here, everything in this gold color are rhamboid or euhedral crystals that we're seeing. In this case, we have a very narrow peak here. That is, these are actually rhamboids, so this is backwards. So I apologize for that. These are all rhamboids in both of these cases. There are 2 little euhedral crystals that are right here. This is diagnostic for open system behavior. There's just one value. All the pyrites have the exact same value. So this is maximum biological fractionation. In an open system, there's no distillation. This is a textbook example of what you'd expect in a closed system where you have distillation, large fractionation by biology, but poor water evolution gives you a pattern like that. So this is clear information about the process that's going on here. We know what biology is doing. We know open versus closed system. However, we see in a lot of those aberrant samples where pyrite particularly gets very heavy, secondary textures. Again, we have these rhamboids that are very depleted. In all cases, there are roughly about -50. This is the biological fractionation. That doesn't change. So as we go through these glacial, interglacial cycles, biology is not changing at all in terms of the isotopic fractionation. They all have differing amounts of open versus closed system. But in some of these cases, we get this huge gap between the isotopic composition of early formed pyrites and, by inference, those that form much later on at the tail end of distillation. And like I mentioned briefly yesterday, this is really important because if we just look at, say, the bulk value of this sample here, which is about +7, that's +7, there are no actual pyrites that form at +7. There's a suite of very depleted values caused by one process. There's a suite of very enriched values caused by another process. Their average is the same as that bulk value. So we know the method works. We know we're not having an analytical artifact. But mechanistically, there is no process, biological or environmental, that gives rise to these bulk values. And that's what people have traditionally done. That's what that burner model does, is you take a bulk value and you try to infer something about sulfur cycling based on that value. And that's why it can work. It works here because there's only one value. But in the vast majority of cases, that does not work and you're led astray. And I want to point out some of these values can be incredibly heavy, up to plus, the bulk value here is about +100. We actually have no idea how you make bulk pyrite that heavy. And it looks like it's caused by one process and then even another process that's going on. So we're still not there in terms of our understanding. But this kind of approach is how we're going to get there. So if I come back in a couple of years, hopefully we'll be able to have answered that. So there are some key take-home messages just based on this approach. These bulk values often average over distinct populations, isotopically distinct, petrographically distinct, that are forming in different environments corresponding to different levels of supersaturation and probably different microbial and geochemical processes. This bulk value in most cases is controlled by sedimentation rate, organic carbon loading, and in the case of pyrite formation, iron availability. We're obviously not able to form pyrite if we're just producing the sulfide, but no iron. These anomalous values seem to be controlled by non-steady state deposition, and possibly enhanced associated with maybe anaerobic oxidation of methane. This is where we're really trying to mechanistically understand that. I think it's really critical to integrate this petrographic, sequence stratigraphic, and SIMS approach to understand the factors that give rise to biology. Once we do that, we can then say things that are really powerful about what's happening on a much bigger scale. Right? So if we take this framework based on microscale observations of individual samples, we can then try to extrapolate. All right, so for doing that, we started out with this microbial model that Itai and Boz put together about a decade ago, looking at how microbes fractionate sulfur isotopes during sulfate reduction. I had a nice detailed model about sulfate going into the cell and getting reduced. And so there's a good framework for understanding the biological controls. We then integrated that with data about sedimentation rate, bottom water oxygen, temperature, organic carbon loading, and sedimentary porosity, things that we think matter for relating microbial activity and diffusion. And we're able to predict the biological fractionation during sulfate reduction, which is mostly invariant, and then the isotopic offset between sulfate and pyrite, which is a lot more variable. And this is variable because of this open versus closed system. Shallow water environments dominantly have much smaller offsets between sulfate and pyrite. This is in no way caused by changes in microbial fractionation. This is all caused by open versus closed system. Or so I'm trying to convince you. This is a comparison of modeled data versus observations. So this is a plot of biological fractionation versus the offset between sulfate and pyrite. Blue data are models for deep environments and shelf environments. The red data are observations from deep and shelf environments. And so hopefully what you can see is that these tend to agree fairly well. So this gives us a lot of confidence that this framework that we have, which again is just based on this sort of microscale analysis, is something that can explain most of the variability that we see today. And again, in both cases, we have this data here only for the model, because we don't actually know what the biological fractionation is in modern settings. But this is showing the predicted values from the model, essentially invariant around very high values. A lot of this variability is caused by temperature change, which changes that equilibrium value. So we're really excited about this. Where do we go from here? This is a compilation of isotopic data through Earth history, where these red dots are sulfate values, these blue dots are pyrite values. We can combine these together to get this offset, this sulfate pyrite offset through time from the Archean to the modern. And that's what this figure shows. And hopefully you can see there's this trend towards increases in this fractionation over Earth history, and also a change in spread from very small fractionations in the Archean through to larger and more variable fractionations today. This has been observed before. This is a nicer compilation, but this pattern has been observed before. And it's been interpreted to reflect small biological fractionation due to sulfate limitation and increasing biological fractionation as sulfate increases in time. That is not the right way to think about that. That contributes a small amount, but it's a very minor amount. This is looking at the change in microbial fractionation as a function of sulfate concentration on a log scale. We're at 28 millimolar today. And you'll see that there is some decrease in fractionation as sulfate values go low, but it's still pretty darn high. And we're showing a model for both modern levels of oxygen and 1% oxygen to try to be better, more relevant for early on in Earth history. And oxygen doesn't matter that much for this change in microbial fractionation. It matters slightly, and that's mostly because if there's less aerobic respiration, there's more organic matter available for sulfate reduction, and that enhances rates of sulfate reduction, and so it's a slightly smaller fraction. On this side, again, log sulfate availability, and then the isotopic difference between sulfate and pyrite. You see here a much stronger signal as a function of sulfate. And this again is not about changes in the microbial fraction. This is about the ease of which you go from an open system to a closed system and how that depends on sulfate concentrations. The lower the sulfate is, the more easily you can move down this trajectory. And here you also see at very low values of sulfate, a much stronger impact for changes in oxygen concentration. Again, that is not changing the biological fractionation, that's changing this offset between sulfate and pyrite. And that's because of changes in the position of the chemocline. If you get rid of oxygen, which again regulates open system or closed system. So I think we can take these very small scale measurements and actually extrapolate them in a pretty major way to understand what goes on for modern systems in terms of both shallow ocean and deep ocean, and then how these patterns have changed over Earth history. So I'm pretty jazzed about that. I agree. And so again, I just want to leave you here with thoughts. Depositional environments really regulate these bulk pyrite values. Understanding the controls here allows us to separate and identify biological and sedimentological processes that happen today and how they've changed over history. And given this context, we need to go back and look at the chemostratigraphic records that everyone has generated for the last several decades, myself included, and reinterpret them in light of this. So with that, I'm going to stop talking and answer any questions.
At the beginning of your presentation, you said this kind of method can be used not only for this type of study, but other types of chemical analyses. Have you seen some particularly interesting examples of such?
So we have started to do this a little bit with things like iron speciation. We have started to do this with things like carbon isotopes, where you see pretty large variations associated with multiple generations of cements, both during early and late stage diagenesis, that are often hard to tease apart by traditional methods, where you'll either take a bulk sample or your micro drill, but you're still often integrating many different phases. So I think those are the ones that we've poked at, but I would imagine it's pretty applicable.
In your last thought there, the major changes that you were seeing on the right-hand side are that it's essentially related to the sulfate availability and how quickly you go open and close.
Exactly.
I guess are there other nutrients in a sense that feed pyrite production that can also play a role in, like, is iron availability or organic matter availability? How do those factors play into all of this? Is it all just sulfate availability?
Yeah, so that is a great question. So the scatter here is running this with. Thank you for asking. Running this with the range of TOC. I don't have it plotted here, but iron porosity. So this is sort of taking the distribution of these parameters today, and then saying, what would that look like if we just changed to oxygen or sulfate? And so that's what causes this variability here, is differential organic carbon availability, differential iron availability, changes in porosity, changes in sedimentation rate. And so that gives us this range based on the present day variation. And so we could go in and look specifically at, as a function of iron availability, what does this look like? So instead of having an axis here, which is sulfate, just zoom in on these 10,000 measurements and say, let's plot that against iron availability. What does that look like? Let's plot that against organic carbon availability. What does that look like? So that data is in here, and you might expect, it does what you think. The more carbon there is, the more closed system you get, because of faster rates of sulfate reduction. And in terms of iron availability, same thing in terms of regulating where the chemocline is, and also what part of that gets into pyrite. If we run out of iron, we stop making pyrite, and the pyrite record is biased towards the lower values.
Can't see from back here, what is the time scale of that change? Is it 10 million years, 15 million years?
So these, here, this is going from the Archean, the Proterozoic, the Neoproterozoic, Paleozoic, Mesozoic, Cenozoic. So these are, hundreds of millions of years intervals. This is modeling the values that we would expect just at different sulfate concentrations. We think sulfate concentrations went from here in the Archean and then migrated to these values over the Phanerozoic. probably and changes that took tens to hundreds of millions of years to sort of move in this direction. But that is just sort of reinitializing the model at different starting sulfate
So
concentrations.
the scale is hundreds of millions of years.
Yep.
So then that begs the question of what Earth process could make such a change?
Photosynthesis?
You mean a linear change over time? But
I mean, we think that we start out with very low sulfate and a no oxygen world in the Archean. With the onset of oxygen in the Proterozoic, we start to build up a sulfate reservoir slowly. And then by the time we get to the Phanerozoic, we have records of sulfate that starts out at maybe 5 millimolar. And then marches up to 28 millimolar today. And that's thought to be driven primarily by changes in photosynthetic output versus organic carbon burial and sulfate burial versus pyrite burial as the two dominant.
and is there a geochemical signature of photosynthesis that allows you to support that statement that has been steadily increasing, for example?
There are geochemical data that allow us to show that sulfate is increasing by looking at pore water sulfate that's preserved in evaporites, so in fluid inclusions and evaporites. So there is independent data to show that Earth has been moving between these spaces. Whether it's due, there's no direct linkage that we can point to that says, there's more photosynthesis here that caused.
So there is not a geochemical signature that you know of where one could independently say that photosynthesis is steadily contributing to this signature.
No.
Okay. So that's where we should be looking. Independently, we should be looking for some biological signature.
But why wouldn't it just be buildup of organic material over time and then bury it, as you said, as opposed to a change in the rate of photosynthesis?
Okay, well, that might be a way of connecting to the signature of photosynthesis, organic accumulation.
I am incredibly excited for those signatures to be found. So I encourage everyone here to go look for them. I'm gonna be happy with this framework.
I'll ask a question. For this global map you're showing, there's actually a really, I think it's the Baker 2016 or so paper that shows you the global distribution of the depth of the sulfate methane transition
Yep.
zone, and where, I mean, it doesn't only show that, but also where there's none and so forth.
Mhm.
Have you tried to compare the maps and seen if
Yeah, so that's the next thing. So this what we're seeing here is the offset between sulfate and the bulk pyrite that is created by integrating down a column. And for all of this, we could then look at those pyrites individually. And when you go through the sulfate methane transition zone, you get a secondary pulse of pyrite formation that is isotopically enriched. That's at the very tail end of the distillation process, which is why I think that process is causing our aberrant heavy values. That is all folded up in here, and I'm excited to try to extract that.
yeah, I mean, they have an independent model from you guys.
Yeah,
right?
yeah.
So it'd be nice to hear and see where it's not lining up.
That's a good idea.
What hope do you have that the existing literature on sulfide isotopes can be sort of gone back through and scrubbed for, like, these bulk values are meaningful, based on the type of sediment that it was deposited in versus, or does the whole thing have to be rewritten?
I mean, the data are what they are.
But are they accompanied, I guess, by the adequate characterization?
Yeah.
of the sediment?
I mean, in some ways, not much has changed because, people have argued that this pattern, of, low offset in the Archean to high offset today, was caused by, say, low sulfate and low oxygen, and high sulfate and high oxygen. That part's right, just mechanistically it's wrong. It's not wrong because it's not how people thought about it in terms of biological controls. It's more about diffusion control. Where I think we struggle is, if you look in the very early records of isotopic geochemical data, there is often a very rich description of the rocks from which these samples came from that talked about lithology, facies, and depositional environment. And so for those, you can actually go in and interrogate those samples in light of this sort of different framework. There's sort of an unfortunate period of geochemistry from maybe the late 90s to today, where often people don't pay attention to the facies or the lithology or the depositional setting. And so that makes it really hard to then place values in the kind of framework that I think is necessary to infer. We could try to invert that situation, and I could say, okay, this is a bulk value of this. I'm going to infer this is your sedimentation rate, or this is the environment. But it would be really great to have that data independently so that we could sort of use that as a proof or to support that.
So how does your carbon isotope record of carbonates from your Italian stratigraphies look like? Have you done any work on that? Or has somebody where you see these like heavy values that are not model predicted?
Oh, in terms of looking for AOM type signals, we do not have that data yet. There is often a barium enrichment, which makes me think that maybe AOM is involved, but we don't have the detailed carbon isotopes.
I have
That's too
detailed
many.
questions. I'd rather not subject to. But I'm happy to, I'm happy to answer, ask.
You can even answer that.
Or try, but a lot of the sulfur you see preserved in sediments is not due to simple progressive accumulation of sulfur. A lot of it's been represents residual sulfur following extensive reoxidation processes. And so I'm wondering what you think the impact of oxidation is on this whole business, because when you get a more closed system, you also get less reoxidation. When you get a more open system, you have much more reoxidation.
So I will say that the way that this model is set up is assuming constant deposition and allows for reoxidation. Where I think it's really important to go forward is to look at non-steady depositions and sort of whole scale reoxidation kind of processes associated with that. That is really hard to integrate into this particular model framework. I think that's going to be important for explaining these aberrant. I'm gonna. Interglacial, glacial, interglacial. These aberrant values, it's up to plus 100, this is up to plus 50. These have to be associated with some I think, some non-steady deposition process associated with reoxidation, reduction, reoxidation, reduction. And so I think that process needs to be incorporated into this framework. I'm not sure how to do it on the modeling side, but from a sedimentological side, we're gonna go back to this section in the summer and really do a detailed characterization to try to identify and set structures that can shed light on processes that may have been happening here. I think that's gonna give us a shift towards heavier values because of enhanced sulfate reduction rates with smaller fractionation. And maybe the removal of depleted values during sort of an erosion event. That's gonna both shift to heavier and more variable values. Like, that's my intuition.
Well, we can pick up more of this later. I don't want to subject.
I think. Do we have any more student questions? No? Okay, let's thank Dave from.